Every living organism needs energy and nutrients to survive and grow, but they don't acquire it in the same way. Some absorb sunlight to power their processes, while others depend on organic and/or inorganic chemical compounds. Oxygen is a key player for many species, but not all need it to thrive, and some just require small amounts. Carbon is a fundamental element that serves as the backbone for essential macromolecules, such as carbohydrates, proteins, lipids, and nucleic acids. This makes a reliable carbon source vital to all living organisms. In today's lecture, we'll explore how eukaryotic and prokaryotic organisms are classified based on where their energy and carbon sources come from, aka their energy and carbon source. We'll also discuss the mechanisms behind energy-yielding processes such as cellular respiration, fermentation, and photosynthesis. You'll learn where these reactions take place, how they operate, and why microbes adopt different metabolic strategies depending on their environment. So by the end of this lecture, I hope that you'll gain knowledge and understand how energy moves through these systems and how switching between one metabolic process to another gives a microbe a survival edge in its changing environments. Let's begin by classifying organisms based on their source of energy and then by their source of carbon. Organisms either get their energy from the sun, referred to as light energy, or from various chemical compounds known as chemical energy. Those that capture energy from the sun are called photoautotrophs, while those that rely on chemical compounds are called chemotrophs. Chemical compounds containing carbon are categorized as either being organic or inorganic. A compound is considered organic when it includes both carbon and hydrogen atoms. Examples include our macromolecules, the carbohydrates, proteins, lipids, and nucleic acids. They all contain carbon and hydrogens. In contrast, the inorganic compounds may or may not contain carbon, but they do not contain hydrogen. For the purpose of defining an inorganic carbon source, we're specifically referring in this case to compounds that do contain carbon, hence a carbon source, but lack hydrogen. In this case, our inorganic carbon source will be carbon dioxide, which is CO2. Thus, organisms that use organic molecules like carbohydrates, proteins, and lipids as their carbon source are classified as heterotrophs. And in contrast, those that rely on carbon dioxide, the inorganic compound, for their carbon source will be called autotrophs. In combining the energy and carbon sources, we now classify organisms in 4 primary metabolic categories: the photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs. In this lecture, I'll cover cellular respiration, fermentation, and photosynthesis with emphasis on the photoautotrophs during photosynthesis and chemoheterotrophs for cellular respiration and fermentation. As you learn about the 3 eukaryotic microbes, the protozoa, the algae, and the fungi, you'll see that they'll group among the photoautotrophs and the chemoheterotrophs. But in contrast, when we talk about the prokaryotic microbes, such as the bacteria and the archaea, these guys will be spread out among all 4 metabolic categories. Now, cellular respiration, fermentation, and photosynthesis are key metabolic pathways that either construct or make or decompose or break down complex molecules through a series of enzyme-mediated chemical reactions. Metabolism is that combination between the catabolic and the anabolic reactions. Sometimes catabolic reactions are called catabolism, and anabolic reactions are called or referred to as anabolism. Now, catabolic reactions are metabolic processes that are going to break down large complex molecules into several smaller ones by breaking down covalent bonds and releasing electrons. In doing so, they're going to release energy in this process. So therefore, these reactions are typically referred to as exergonic reactions, meaning they produce energy that's released. This energy that's released is then captured into a form of usable energy known as ATP. Whereas the anabolic reactions are metabolic processes that build complex molecules from several smaller ones. These reactions need energy, and they're going to need it in the form of ATP. So they're going to use that energy to build covalent bonds, to assemble these larger structures, and we're going to define them as being intergonic because they use that energy. Before I start to discuss these different metabolic pathways, it's important to understand how energy is converted, either from light energy to chemical energy, as in photosynthesis, or from one form of chemical energy to that of another during cellular respiration and fermentation. So let's start with the key players: the enzymes, cofactors, coenzymes, and ATP. Enzymes are often referred to as biological catalysts because they accelerate chemical reactions that would otherwise take years and years to occur. They do this by lowering their activation energy that's needed for reactions to occur. That means the activation energy that's needed to either create or destroy covalent bonds. This activation energy is the minimum amount that is required for that reaction to occur. Now, enzymes work with substrates. Substrates are molecules that bind to their active site. So I have here that substrate binding occurs when one or more molecules nestle into the enzyme's active site. So this area here is the active site of this blue enzyme. It's very reminiscent of a lock and a key mechanism. So this analogy reflects to the specificity of the interaction where the substrate or substrates and the active site possess complementary shapes and charges. So you can see that the shape of the active site is reciprocal to the shape of this particular substrate shown here in the top left corner. In this upper reaction, we have a single substrate. It's going to align precisely with this enzyme's active site. The enzyme will then go through a slight or a subtle conformational shift, which will facilitate the breakage of a covalent bond within that substrate, and will change that substrate into two products. The resulting formation of these two product molecules and the release of energy from that substrate will cause the products to no longer match with that of the active site of the enzyme. So they will leave the enzyme as products. Again, this process exemplifies an exergonic catabolic reaction where a large molecule is broken down into two smaller molecules, releasing energy. Thus, the resulting products are said to have less energy than the incoming substrate had. In contrast to that, let's look at the lower reaction, which involves two distinct substrates, and they're going to bind simultaneously to the enzyme's active site. The enzyme again will undergo a slight conformational change, thus promoting the formation of a covalent bond, and this requires energy to occur. So the substrates bind and a covalent bond forms, producing a new product. This resulting product contains more stored energy than its substrates had, indicating that energy has been invested in this bond formation. And as in the previous reaction, the products no longer fit in the active site and is thus released. This illustrates an endergonic anabolic reaction where two smaller molecules were joined and formed a larger complex, more energy-rich product. Now, in both cases, these enzymes can interact with their substrates without any assistance, and this is not always the case. Sometimes an enzyme needs a cofactor or a coenzyme in order to form that active site and in order for it to be able to interact with its substrate. So as an example, I'm showing you a picture here in which these coenzymes become catalytically active when they bind to either a cofactor and or a coenzyme. In both cases, they will contribute to the proper formation of the enzyme and the function of that active site. So let's talk about cofactors versus coenzymes. Cofactors are typically small, positively charged metal ions, and I'm giving you a few examples here. I have iron, magnesium, and zinc. These help stabilize the enzyme's tertiary structure and can also assist in the substrate's binding by neutralizing the charge within this interaction, because notice they have that positive charge. Coenzymes are much larger. They're larger organic molecules that associate with the enzyme to complete the active site, as shown here. Examples of coenzymes will be NAD+, NADP+, and FAD, and you'll see these in today's lecture. These coenzymes will act as electron and protein carriers. So they're going to extract electrons from the substrates and carry them over to other molecules. These transferred electrons are ultimately used to generate high-energy ATP molecules, which will power cellular processes. So when discussing these coenzymes in context of redox reactions, it's essential to understand the terms oxidation and reduction. When a molecule, atom, or ion is considered oxidized, it's because it lost electrons. So coenzymes such as NAD+, NADP+, and FAD are currently in their oxidized form because they have not yet accepted electrons. So we're going to say that they're electron deficient. Conversely, when these molecules gain their electrons, they now become reduced. Upon accepting electrons and hydrogen ions from their donor molecules, NADP+, NAD+, and FAD are now transformed into their reduced forms. In order to maintain efficient metabolic functions, coenzymes have to constantly alternate between their oxidized and reduced forms, and this ongoing redox cycling will help facilitate the movement of high-energy electrons that come in through nutrients, through various metabolic pathways. It will help break down carbohydrates, proteins, and lipids, and ultimately release that energy during these electron transfers, and ultimately harness that energy to make ATP, which is the cell's principal energy. So the energy will come in through our nutrients, and we will store that energy into a usable form of ATP. This is a picture of ATP, which is also called adenosine triphosphate. Structurally, ATP has a adenine nitrogenous base. It has a ribose 5-carbon sugar and 3 phosphate groups. The chemical bonds that link these phosphate groups together have high energy, specifically the bond between these 2 phosphate groups shown here. When this terminal phosphate group is released, it forms the molecule ADP, adenosine diphosphate, and releases a lot of energy. That energy will drive essential cellular activity, such as muscle contraction, active transport across the cell membrane, or the biosynthesis of macromolecules. So it's important to sustain life in all organisms. So these cells have to constantly generate ATP from ADP, and it does that through cellular respiration in heterotrophs or photosynthesis in autotrophs. So now that we have the basics, let's begin to discuss cellular respiration. We'll follow the flow of carbon and electrons to understand where the atoms go, how energy is released, and how matter is recycled. This approach is important because the flow of carbon is tightly linked to this high-energy electron transfer. When carbon-to-carbon bonds or carbon-to-carbon hydrogen bonds are broken, high-energy electrons are released. These electrons are then captured by these coenzymes, which I mentioned, which was NAD plus and FAD, to form the reduced forms, which is then is used to make ATP. So by following the path of carbon, we can better understand why these specific steps in cellular respiration generate ATP, NADH, or carbon dioxide, and how these outputs contribute to the cell's overall energy economy. Now, many organisms, including us, humans, rely on cellular respiration to extract energy from the foods that we eat, such as carbohydrates, proteins, and lipids, and to generate the precursors for building essential macromolecules like proteins and lipids. In most discussions of cellular respiration, we generally focus on glucose, and that's largely for the sake of simplicity. Glucose is that simple sugar. It serves as a primary energy source for a wide range of organisms, meaning that every organism that we know can break down glucose by cellular respiration. It's easily metabolized through glycolysis, the Krebs cycle, also known as the citric acid cycle, and oxidative phosphorylation, which is the electron transport chain and chemiosmosis collectively. In animals, glucose is readily absorbed from the bloodstream. In plants, it's produced by photosynthesis and funneled into the energy pathways. Whereas proteins and lipids, on the other hand, require a few extra steps. Lipids, also known as fats and oils, are broken down into glycerol and their fatty acids. Glycerol enters glycolysis and is converted to a glycolytic intermediate, as shown here. Whereas their fatty acids are broken down into acetyl-CoA, which then enters the Krebs cycle. Proteins, on the other hand, are large polymers of amino acids. So these amino acids are cleaved, their peptide bonds are broken, and the amino acids are then deaminated. That means we remove their amino groups, and the resulting carbon backbone is converted to various intermediates that can enter glycolysis, they can enter the Krebs cycle, or the intermediate steps. So while these alternative pathways are biologically important, they involve more complex steps and are slower, and they vary more across species. This makes glucose metabolism the most straightforward and widely applicable model for studying cellular respiration. So this is the purpose or the reasoning where the rest of this lecture will focus on the catabolism, the breakdown of carbohydrates in the form of glucose. But I'd like you to also understand that during a normal metabolizing cell, we also do catabolism of proteins and catabolism of lipids. So let's delve deeper into cellular respiration. Cellular respiration occurs in two distinct modes, that is, aerobic or anaerobic. During aerobic cellular respiration, the process occurs in the presence of oxygen, whereas in anaerobic respiration, this takes place in the absence of oxygen. Despite these differences, both pathways share a common sequence of core biochemical events known as glycolysis, the intermediate step, the Krebs cycle, the electron transport chain, and chemiosmosis. The critical distinction lies in the terminal electron acceptors that utilize the electrons that are transported along the way. I'll elaborate on these acceptors when we reach the stage of discussion, and that will be during the electron transport chain. So let's start with that of glycolysis. Now, glycolysis takes place in the cytoplasm of both prokaryotic and eukaryotic cells. It involves 10 enzymes, and you can see them here in blue. Each of these enzymes is also represented as 10 different steps, shown as these yellow circles with the numbers. Now, these 10 steps are to split glucose, a 6-carbon molecule, into 2 pyruvic molecules, which each contain 3 carbons. Remember, we're going to follow the flow of carbons and the flow of electrons. During glycolysis, some carbon-to-carbon or carbon-to-hydrogen bonds will be broken with the aid of enzymes and NAD+, the coenzyme. These high-energy electrons will be released and captured by NAD+, and reduce NAD+ into the form of NADH, which will then carry the electrons to the electron transport chain. Remember, the electron transport chain occurs later on during cellular respiration. Recall that NADH is the reduced form of the coenzyme NAD+. That will transfer. Now, steps 1 and 3 are called energy investment steps because energy from ATP, remember, the bond between the terminal phosphate groups is high energy. So we're going to release that energy from ATP and transfer it to glucose in step 1 to make glucose 6-phosphate, and transfer more energy to fructose 6-phosphate to produce fructose 1,6-biphosphate. In steps 4 and 5, fructose 1,6-biphosphate will be cut in half to produce 2 different molecules, one of dihydroxyacetone phosphate and the other one of glyceraldehyde 3-phosphate. Now, the cell can't use dihydroxyacetone phosphate as it is, so it has another enzyme which converts this molecule to a second molecule of glyceraldehyde 3-phosphate. So I'd like you to understand that one molecule of glucose will undergo 5 reactions to generate 2 molecules of glyceraldehyde 3-phosphate. Both of these glyceraldehyde 3-phosphate molecules will be oxidized, as shown here, meaning oxidized, they are going to lose electrons. They're going to transfer their electrons and protons, hydrogen protons, to NAD+ to form NADH. So this is a redox reaction, where one molecule is being oxidized, another is being reduced. These NADH molecules will deliver these electrons to the electron transport chain. This takes us to steps 7 through 10. So steps 7 and 10 are energy payoff steps because energy is extracted in the form of ATP. So notice that 1,3-bisphosphoglycerate is losing a phosphate group, it's donating it to ADP to make ATP. So we're extracting energy. And again, phosphoenol pyruvate is going to lose its phosphate group to make pyruvate, and that phosphate group will attach to ADP to make ADP. Now remember that we had 2 molecules of glyceraldehyde 3-phosphate, so these energy payoff phases occurs 2 times, 1 time each for each glyceraldehyde 3-phosphate. So we're generating 2 molecules of ATP here times 2. So ultimately, we've gained 4 molecules of ATP. But recall that we used up 2 molecules of ATP during the energy investment phase. So therefore, we say that we have a net gain of 2 molecules of ATP. That accounts for the loss in the initial steps. Now, this is a lot of information, but this is what I want you to know. I want you to know that the energy investment phase, steps 1 through 5, and these steps of glycolysis, 2 molecules of ATP are consumed, and so I'm showing you that right here in the summary. And that glucose is ultimately generates 2 molecules of pyruvate. During the energy payoff phase, 4 molecules of ATP are made from ADP, but remember the net gain is that of only 2. And lastly, I'd like you to know that in step 6, that we had a redox reaction in which glyceraldehyde 3-phosphates were oxidized and generated 2 molecules of NADH. So I'd like you to know what goes into the reaction and what goes out. I'd like you to look at this 10 steps of glycolysis and be able to answer a few questions on the exam based on that figure. So as mentioned, that 1 molecule of glucose will produce 2 molecules of pyruvic acid. These 2 pyruvic acids or pyruvate molecules will then move on to the intermediate step. This is the intermediate step right here. This step occurs in the mitochondrial matrix of eukaryotic cells or in the cytoplasm of prokaryotic cells. Remember, they lack organelles. During this step, a carboxyl group is removed in the form of carbon dioxide from each pyruvate and releases these carbon dioxides as waste. The remaining acetyl fragments, which is a 2-carbon molecule, is then oxidized. We're going to donate electrons to NAD+ to make NADH. And at this moment, a coenzyme A will attach to acetyl group. This is the CoA or the coenzyme A molecule in black, and this is the acetyl group that's represented by the carbon double bonded O, CH3. So just like I expected for you from glycolysis, I'd like you to be able to look at this figure and give me the results of what happens during the intermediate steps. So I'd like you to understand it enough that you can answer questions. So the net result here is per 1 molecule of glucose. That means that we have 2 molecules of pyruvic acid that were generated. We are going to release 2 molecules of carbon dioxide. We're going to produce 2 molecules of NADH and 2 molecules of acetyl that were covalently linked to coenzyme A. Now this carbon dioxide is released. The NADH that's produced will go to the electron transport chain, and the acetyl-CoA will go on to the Krebs cycle, which is the next cycle. So here's my acetyl-CoA molecule. Now acetyl-CoA is a high-energy compound. It is going to donate its acetyl group to the oxalic acid. That's in the first step of this Krebs cycle. And in doing so, it's going to generate citric acid. This Krebs cycle is sometimes referred to as the citric acid cycle because it makes citric acid, as well as the TCA cycle. And it's going to consist of 8 enzymes or 8 steps that take place in the mitochondrial matrix of eukaryotic organisms or the cytoplasm of prokaryotes. The result of the Krebs cycle is to completely oxidize the molecules that came in with glucose. That means that we'll extract as many electrons that are possible. So an overview of the Krebs cycle is that 1 molecule of glucose, remember, produced 2 pyruvic acids, so we're going to have 2 acetyl-CoAs. So that's what this 2 rounds represents. So if we do it 1 cycle, acetyl-CoA is going to combine with oxaloacetate, as shown here, and it will produce a molecule of citric acid. The citric acid is going to be rearranged and oxidized and will release a molecule of carbon dioxide. And here's the oxidation step as shown by NAD+ to NADH. These electrons are going to be transferred to NADH, and we're going to do this redox reaction again further down. Again, and this time with FAD to make FADH2, and a fourth time with NAD+. We're going to generate 1 molecule of ATP per round, so total 2 molecules of ATP. When all is done, that will bring us back to the molecules of oxaloacetate. It will be regenerated, and it will be ready to interact with another incoming molecule of acetyl-CoA. So for every 1 molecule of glucose, we will have 2 cycles of the Krebs cycle. So we will have 2 incoming acetyl groups that will generate 4 molecules of carbon dioxide. 1, 2, times 2. We will generate 6 molecules of NADH. So 1, 2, 3, again, times 2. And 2 molecules of FADH2, 1 times 2. And 2 molecules of ATP. So before we move on to the next phase, that is the electron transport chain, let's go back and see what we just accomplished. So we had 1 molecule of glucose that produced 4 molecules of ATP, 2 net in glycolysis and 2 in the Krebs cycle. In combination of the intermediate step and the Krebs cycle, we generated 6 molecules of carbon dioxide. And also, in total, we've generated 10 molecules of NADH and 2 molecules of FADH2. It's these molecules that will go on to produce the majority of ATP. Remember, so far we've only made 4. We want to generally make about 36 to 38 molecules of ATP. And this will happen in the next steps. So let's get back now to those NADH and FADH2s that were made during the initial steps of glycolysis, the intermediate step in the Krebs cycle. Their job was to carry those electrons to the protein complexes shown right here. Now this happens in the mitochondrial inner membrane of the eukaryotic organisms or in the cell membrane of prokaryotic organisms. When NADH and FADH2 donate their electrons, the electrons will move through a series of membrane-bound carriers. This includes these complexes 1 through 4, as well as cytochromes and ubiquinones. And as the electrons are cycled over carried from one complex to another, they will slowly lose some of their energy. That energy from these electrons are released during the electron transfer and used to pump out hydrogen ions. So for instance, complex 1, while it's reduced, it actively pumps out hydrogen protons from one side of the membrane to the other. And then when it quickly loses electrons to reduce another molecule, it stops the pumping. So these hydrogen pumps are activated only when they are in the reduced mode. This concentration gradient also forms an electrical gradient because it has a net positive charge. In aerobic cellular respiration, these electrons are ultimately bound or combined with oxygen molecules. So we say that oxygen during aerobic cellular respiration are the final electron acceptors. Once that happens, they combine to hydrogen protons and they form water, which keeps the electron transport chain flowing. Now, that's not the whole picture. We're going to take this part where the oxygen acquires electrons up to the point where it becomes water, and we're going to show more detail. So during aerobic cellular respiration, oxygen is converted into superoxides. Organisms that are capable of aerobic cellular respiration have enzymes that can convert these superoxides into hydrogen peroxide and then further into water and oxygen. So these enzymes are superoxide dismutase, peroxide, and catalase. If an organism does not have one or more of these enzymes and are metabolizing the presence of oxygen, the superoxides will build up and kill the organisms. Some organisms, such as certain bacteria and archaea, these are the prokaryotes, they either lack the proper molecules to transfer these electrons to oxygen, or they lack these enzymes needed to convert the superoxides back to hydrogen, back to water and oxygen. In these cases, these prokaryotes have adapted to perform anaerobic cellular respiration. Anaerobic cellular respiration is respiration in the absence of oxygen. So in the absence of oxygen, these molecules can use nitrates, as shown here, or sulfates to serve as their final electron acceptors. In this case, if they use nitrates, nitrates will be reduced in the form of nitrites, and this nitrite is then expelled from the cell and used by other organisms as a source of nutrients. So in either case, whether it's aerobic or anaerobic cellular respiration, the electron transport chain pumps out hydrogen protons from the mitochondrial matrix into the intermembrane space of eukaryotes, or the cytoplasm to the periplasmic space of prokaryotes. The only difference is their final electron acceptor. Know that the function of this electron transport chain is not to make the extra ATP molecules that are needed, but instead it is there to reoxidize or recycle NADH back to NAD+, and FADH2 back to FAD. This helps replenish the cell's storage of NAD+ and FAD to help them continue cellular respiration. That's the first function of the electron transport chain. The second is that they create this hydrogen proton gradient, or this electrical gradient. This gradient is going to fuel the production of ATP synthesis during chemiosmosis. So chemiosmosis is the event where hydrogen protons flow back down across their gradient through the ATP synthase, which is this membrane-embedded enzyme. And as they do so, they help rotate the enzyme or activate the enzyme to drive the conversion of ADP back into ATP. So keep the electron transport chain separate from chemiosmosis. Now this brings us to the next metabolic pathway, and that is of fermentation. Fermentation is a partial form of cellular respiration that includes only glycolysis. Unlike aerobic or anaerobic cellular respiration, fermentation does not involve the intermediate step, the Krebs cycle, the electron transport chain, nor chemiosmosis, but instead it relies solely on glycolysis to generate small amounts of ATP, and then uses the organic molecules pyruvate or its derivatives as their final electron acceptor to regenerate more NAD+, allowing glycolysis to continue. Now this figures to show you that various microbes can carry out different forms of fermentation, each utilizing pyruvate derived from glycolysis to generate different end products, as shown here. But the primary objective across this pathway is, again, to regenerate that NAD+ to allow glycolysis to continue in the absence of oxygen. For our purpose, we're going to focus on only two types of fermentation, that of lactic acid fermentation and alcohol fermentation. Lactic acid fermentation occurs in certain microorganisms such as lactic acid bacteria and in human cells, particularly our muscle cells under anaerobic conditions. Now, during this process, NADH that is produced during glycolysis is going to transfer its high energy electrons to pyruvate. It's going to reduce it into the form of lactic acid. So if we look at pyruvic acid here that was generated as the end product of glycolysis, he's going to acquire an electron that was donated by NADH. So NADH is going to donate its electron to pyruvic acid. it's going to reduce pyruvic acid into the form of lactic acid. By reducing pyruvic acid, it is oxidized into NAD+. So keep the terms oxidation and reduction straight. In bacteria, lactic acid is typically expelled as a metabolic waste product, but in humans, it accumulates temporarily in our muscle cells, where it's later then converted back to pyruvate once oxygen becomes more available. But again, I'm going to stress this many times, the ultimate purpose of glycolysis is to regenerate NAD+, to ensure that glycolysis can continue and more ATP production is made. That's lactic acid fermentation. Now, alcohol fermentation has an additional step beyond glycolysis. First, that pyruvic acid is decarboxylated. It removes a carbon dioxide and it produces the intermediate step, acetaldehyde. Only then will NADH be reoxidized by donating its high-energy electrons to acetaldehyde and produce ethanol. So this type of fermentation is characteristic of yeast cells. We use yeast such as Saccharomyces cerevisiae to produce our alcoholic beverages for the purpose of producing ethanol. We also use the same yeast, baker's yeast, to produce carbon dioxide to make leavened bread. So different examples of where fermentation is useful. Now, I don't want you to memorize this table. It won't appear on your exam, but the point of showing you this table is to give you an idea of how microbial fermentation affects our lives. So, for example, lactic acid fermentation is used by lactic acid producing bacteria that are used in yogurts. They ferment milk sugars. They produce lactic acid, which gives the yogurt its distinct flavors. I've already mentioned about ethanol fermentation by Saccharomyces cerevisiae, used for our alcoholic beverages or bread. That takes us to our mixed acid fermentation. Now, E. coli and Salmonellas perform this type of fermentation to produce lactic acid, acetic acid, succinic acid, formic acid, carbon dioxide, and hydrogen gas. This mixed acid fermentation is used as a biochemical test in order to identify or to name the key organisms that are pathogenic that cause infection in patients. So it's used as a test to help identify organisms. Lastly, are the propionic acids. Propionibacterium species will perform propionic acid fermentation, which is then used in dairy fermentation to help process the dairy sugars to make cheese. So these are just a few examples of the many types of fermentation pathways. So now this brings us to our third and last metabolic pathway, that of photosynthesis. In microbiology, photosynthesis refers to the light-driven process by which certain microbes, such as cyanobacteria and algae, can convert carbon dioxide into organic compounds such as sugars or carbohydrates, using light energy to drive the reaction. Unlike plants, microbial photosynthesis can vary in its electron donors and whether or not they produce oxygen. So this is divided into oxygenic or anoxygenic photosynthesis. During oxygenic photosynthesis, as an example, cyanobacteria or algae will use water as its electron donor and will release oxygen. I'll show this reaction in more details in the next coming slides. Whereas during anoxygenic photosynthesis, such as purple and green sulfur bacteria, are not going to use water as an electron donor, but instead will use hydrogen sulfide or ferrous iron. In this case, oxygen will not be produced. So that's the main difference between oxygenic and anoxygenic photosynthesis. In either case, these microbes play their essential roles in global carbon cycling in which we'll talk more about in our upcoming lectures on biogeochemical cycles. In photosynthetic eukaryotes, photosynthesis is going to take place within chloroplasts, while in photosynthetic prokaryotes, the process will occur along specialized infolding cell membranes. Photosynthesis is typically divided into two phases, the light-dependent reactions and the light-independent reactions. So I'm going to begin by explaining the light-dependent reactions. From this point on, I'll mostly focus on oxygenic photosynthesis, and because of that, you're going to see that oxygen will be produced. Remember, under oxygenic photosynthesis, that water is the electron acceptor and oxygen is produced. This figure in the lower left corner provides a more detailed look at this light-dependent reaction. This represents the membranes of either the thylakoids in chloroplasts for eukaryotic organisms or the cell membrane folds in prokaryotic organisms. So let's start with the light-dependent reaction. The first step is that of sunlight. Sunlight is going to energize this photosystem number 2, PS2, which is a transmembrane protein complex that's embedded inside the membranes. This energy will initiate a photolysis, meaning light energy will be used to break up water. It's going to release oxygen and hydrogen ions, while its electrons are going to be funneled into the electron transport chain in a process that is reminiscent of the cellular respiration that we just went through. Ultimately, the final electron acceptor is NADP+. Remember, in cellular respiration, we saw NAD+. NADP+ is similar, except it has a phosphate group. And once it accepts its electrons, it will be reduced in the form of NADPH. During this flow of electrons, hydrogen ions are going to be pumped across the membrane, will create a proton gradient, similar to that we saw during the electron transport chain in cellular respiration. This proton motive force is going to power ATP synthase, which will make more molecules of ATP by adding phosphate groups to ADP, which is strikingly similar to the chemiosmosis, again, that we saw in cellular respiration. Now this ATP and NADPH that was generated during the light-dependent reactions will be the energy and the reducing powers of the light-independent reactions. So this light-independent reactions, also known as the Calvin cycle, occurs in the cytoplasm of prokaryotes and in the stroma of chloroplasts in eukaryotic cells. This cycle will involve three key phases. The first is that of carbon fixation. Carbon will attach to a 5-carbon sugar called RUBP, and it will form phosphoglycerate. The second phase is that we will reduce phosphoglycerate. So ATP and NADPH are used to convert phosphoglycerate into G3P, which is glyceraldehyde 3-phosphate, a 3-carbon sugar. Recall that we just talked about glyceraldehyde 3-phosphate that was being made or formed during glycolysis of cellular respiration. The third phase is that of regeneration. So some of this glyceraldehyde 3-phosphate will be used to regenerate more RUBP, while the remaining will be used to synthesize sugars such as glucose and other carbohydrates that are essential for cellular metabolism. So I hope that you'll see the connection between the light-dependent and light-independent. I'd like you to see the flow of energy that came in through the light energy, was converted into energy through NADPH and ATP. That energy was used to activate the Calvin cycle. We lost energy and created ADP and NADP+, which is then regenerated through another cycle of light activating the light-dependent reactions. and the process continues. So as you can see, that metabolism encompasses all chemical reactions within a cell, divided into two complementary processes, that of catabolism and anabolism. Catabolism is the breakdown of molecules that release energy. As an example, we take a large molecule of glucose and we break it down into two smaller molecules, carbon dioxide and water, and we've released energy in the form of ATP. Whereas anabolism will make large complex molecules from simpler ones and requires energy to do so. In this case, we take the monomers of a protein, amino acids, covalently link them by peptide bonds, which requires energy to make their macromolecules. Some of the key players that were presented here were that of ATP, adenosine triphosphate. It's the universal energy currency. It links catabolic energy release to anabolic energy demanding reactions. We also talked about enzymes, which are the biological catalysts that accelerate reactions by lowering their activation energies. And finally, we talked about coenzymes, specifically NADP plus, NAD plus, FAD. And we talked about coenzyme A. So these organic factors helped enzymes and often transferred electrons or functional groups. So in summary, cellular respiration, fermentation, and photosynthesis are core metabolic processes that manage energy flow in living organisms. Cellular respiration breaks down glucose. It produces ATP with either aerobic respiration or anaerobic respiration. Aerobic cellular respiration uses oxygen for maximum yield, and anaerobic cellular respiration relies on an alternative electron acceptor such as nitrates and sulfates. When oxygen or other acceptors are not available, the cells can undergo fermentation, which allows glucose to continue to generate smaller molecules of ATP, as well as NAD+, and end products such as lactic acid or ethanol. In contrast, photosynthesis captures energy from the sun, light energy, to make organic molecules. You have the light-dependent reactions which generate ATP and NADPH while splitting water and releasing oxygen. And then the light-independent reaction, also known as the Calvin cycle, uses this energy to fix carbon dioxide to form sugars. Together, these pathways illustrate the dynamic balance between energy capture, transformation, and utilization in cells. And with that, this will conclude the lecture on microbial metabolism.